This protocol is significant because it details how to quickly generate large amounts of DNA template for use in cell-free reactions from a small gene fragment received from a synthesis facility. Rolling circle amplification can generate large amounts of DNA faster than traditional cloning, so it supports higher throughput design, build, test, learn cycles from libraries of synthetic DNA. These techniques can have large inherent variability for new users due to the many steps and small volumes required. Careful attention should be given to the technique, and practice makes perfect.
To begin, add all the components from a PCR kit in a single PCR tube. Then add 1 microliter of the resuspended gene fragment. Gently homogenize the mixture by vortexing on a medium setting for 5 to 10 seconds. After programming the thermocycler according to the kit manufacturer's protocol, run the PCR. Upon completion of the PCR, use a PCR cleanup kit to purify the DNA from the reaction mix. In a 1.5 milliliter tube, add the DNA binding buffer and the PCR sample at a 5:1 ratio. Transfer this mixture to the spin column and centrifuge at 16,000 times G for 1 minute. Discard the flow-through. Add 200 microliters of the DNA wash buffer to the column and incubate it at room temperature for 1 minute. Centrifuge the column again and discard the flow-through. Wash the column once more with the DNA wash buffer as demonstrated. After discarding the flow-through from the second wash, remove any remaining buffer by centrifuging the column for an additional 1 to 2 minutes.